I would like to welcome Dr. Suzadenki and one of the coordinators of the Instruct-ERIC Hub, the person that would explain everything to us. And later on, after her presentation, she would answer our questions. Thank you.
Okay, thank you very much. I'll just get my slides up. Okay, I hope you can see my slides. So thank you very much for inviting me to make a presentation to you today. As you've heard, I'm the Instruct Hub coordinator located in the UK, which is where the hub is located in Oxford, UK. And I sort of take an overview of all of the activities that Instruct gets involved in. And I think as we go through the presentation today, you'll see that Instruct has a lot of activities that are ongoing, both internally, so to deliver infrastructure and the various programs that we have ourselves, but also we have a number of collaborative actions and associations with other European research infrastructures and programs as well. So there's a lot going on in Instruct, and I want to give you a flavor of that today. So Instruct-ERIC, a very strange name, I know, but Instruct is the European Research Infrastructure Consortium for Structural Biology research in Europe. And it was formed as an ERIC, which actually stands for, sorry, let me skip that one. ERIC stands for European Research Infrastructure Consortium, which is a specific legal entity that was put forward by the European Union to regulate and construct these research infrastructures to provide services across Europe. So Instruct became an ERIC in 2017. So we have been operating as that legal identity for 4 years. But before that, we underwent quite a long process of establishing the consortium and establishing our procedures for providing access. And we did that through a different legal form, which was as a small company associated with the University of Oxford. But now that we are an ERIC, we have our own legal identity, and that also confers to us some privileges, such as we are tax exempt, and we have open border collaborations with respect to employment and exchange of materials. So being an ERIC is a great thing. We're one of many other research infrastructures in Europe. I think at the moment there are about 55 ERICs of various types, different research infrastructures of this nature in Europe across all domains, from physical sciences, environment, energy, social sciences, right across the board. And I think that there have been 11 more just approved by the European Commission recently. So the number of research infrastructures is growing. But we are the only one in structural biology, I'm pleased to say. So our ERIC is composed at the moment of 15 members, and membership is endorsed at the government level, usually the appropriate ministry that is in charge of science and innovation or something similar, which means that we have a very secure high-level funding for a period of time. And the way that we try to manage our membership is on a 5-year rolling cycle. So we are in our first 5-year cycle of being an ERIC. We've had 4 years so far, and we are now working towards our next 5-year cycle, which will start in 2022. So you can see here the 15 members that we have. They are distributed around Europe, and they include 14 member states and EMBL as an intergovernmental organization. And EMBL joined within the last 12 months. So what is Instruct-ERIC in practical terms? Well, it's a collection of members, most of whom actually host Instruct centers. And the centers is where the infrastructure is located and where services are delivered. And you can see here that in each of the countries or members that have centers, there are a number of specialisms that are made available for researchers across Europe. For example, in Belgium here, you can see that we have the Nanobodies Center, and this is a very specialist center that provides nanobodies and megabodies that are very useful for structural analysis, amongst other things. And additional to that, there is a different research site associated with the center in Belgium that hosts a Robotine platform, which is a protein production and analysis platform. So in each of the members that host centers, there are a number of specialist technologies that are available for access to researchers. And I'll just go through a few of these in a bit more detail. So the services that we offer collectively across all of the Instruct centers, there are about 80 separate services that are available for access. And these fall into three major groups. The first is sample preparation, and this can be protein production, but also not just single proteins. It can be macromolecular complex formation as well. Crystallization is involved in this too, and the nanobody discovery. But it might also include things like labeling that is specific for a particular technique, such as NMR, for example. And then the second category is biomolecular analysis, and this covers a number of technologies, some imaging work, such as super resolution imaging, quite a bit of mass spectrometry, and molecular biophysics technologies like SPR and AUC and various things like that. And then we come to structural analysis, 3D structural analysis. And again, the three major technologies in use there are electron microscopy, various resonance techniques, including solution and solid state NMR, and X-ray techniques. And that is macromolecular crystallography, but also biosacs, so small angle scattering, and more recently, crystallography-based fragment screening approaches as well. So all of these are available to researchers through Instruct. So if we have a look within these categories that I just mentioned, some of the services that are available, you can see here a little bit more detail there. In crystallization, we have macromolecular crystallization, but also membrane protein crystallization in lipid phase, lipid cubic phase. The nanobodies and megabodies, which I mentioned before. And in protein production, there are various protein production platforms that are available from cell-free right through to E. coli, insect, mammalian platforms as well. We also have ESPRI library-based screening available in Grenoble, and as I mentioned, the Robotine platform, which is now available in Belgium since quite recently. And then here are some of the biomolecular analysis platforms that we have available in various types of microscopy, including confocal microscopy, but as I said, super resolution microscopy as well. And then in mass spectrometry, we have native mass spec, but also platforms appropriate for proteomic analysis. And coming soon to Instruct will be 2 new platforms at the UK Centre, one being a hydrogen deuterium exchange mass spec platform that measures the solvent accessibility of protein backbones and allows you to resolve conformational changes in proteins. And the second is mass photometry, which is a method that's been around for a while, but it's a very sophisticated method for measuring the mass of single molecules. And again, that's very useful for analyzing dynamics and ligand binding, for example. And then in molecular biophysics, we have a number of technologies there to characterize proteins and complexes prior to structural analysis. So in the structural analysis, you could choose from electron microscopy, and here we have cryo-EM, including tomography, and other supporting technologies, scanning EM, transmission EM, and importantly, image processing. So in lots of cases, for cryo-EM, you can have some data processing on the fly. And there are also specific image analysis software available for use later on. And then our NMR techniques are very high end, and they include access to Florence's 1.2 gigahertz machine there. But also the technologies available include paramagnetic resonance and relaxometry as well there. And then X-ray techniques, as I mentioned, X-ray diffraction and small angle scattering, but also at 2 of our centres now, high throughput fragment screening based on crystallography approaches. And we also have fragment screening available through NMR methods as well. So our facilities are open to any scientist who has a strong research proposal which could benefit from access to any of our services. The proposals that we accept are all peer-reviewed. So they go out to 3 independent reviewers, and they are reviewed for scientific merit as the first criteria for success. And then if it passes scientific peer review, then the requirements of the user for accessing infrastructure are looked at, and the centres that are most appropriate to provide those services are consulted to look at the scope and what will be needed to fulfil the requirements of the proposal. So Instruct provides the instruments, the infrastructure at the centres to do that work. It also provides expertise from facility staff because they are the ones on the ground running the equipment and advising on the best experimental methods and so on, so that you get the best experience and the best research outputs from the work. And Instruct provides a funding contribution for both the researchers themselves, but also the centres for providing the infrastructure and the time to do the work with you. And Instruct has always supported an integrated approach to structural biology, because we have so many services available, many of which are complementary and can be used in a sort of pipeline configuration. We support that sort of approach. So if you have a proposal that could benefit from more than one technology, then this could be supported by Instruct. So you could do protein sample production, and then you could do some biophysical characterization using our infrastructure there, and then you could go on to do your structural analysis as well. So the full pipeline could be supported, and we're very open to discuss that sort of use. So how do you get access? Well, the first thing that you do is you should go to our webpage and you should register as an Instruct user. So there's a big green button on the homepage saying register. So you have to be registered in our system. So then you can log in, and wherever you see the bright red button that says request access, it's on pretty much every page on the website. Then that's the entry point for you to put in a proposal. As I just said earlier, if you put in a proposal and you are a researcher from one of our members, so any one of our 14 member countries or from EMBL, then your proposal, if it's approved scientifically, will be allocated a funding contribution to support that work. So in most cases, you'll find that the work itself, the access to infrastructure, is free for you at the point of access. It won't cost you anything to do that work. There are some very high-end specialist technologies that, by the nature of their complexity and their cost, if you are looking to do a very big experiment or for a long period of time that would take a lot of our resources, then we would probably have to have a conversation about how you want to manage that work and whether there is some way of doing it so that it minimizes your contribution, but it may not be zero. Okay, so in the last almost 2 years now, we have had to change the way that we've offered our research infrastructures a little bit. And it's because of the travel restrictions and so on, it's become obvious that we can't expect people to visit all of our centres to do the work in person themselves. So over the last 18 months or so, many of our facilities have developed remote access so that the infrastructure can still be used for your experiments, but you can mail in your samples or you can send your samples in some other way so that the work can still be done at the infrastructure and you can receive the data back. And in many cases, that remote access allows you to still run the experiment using a remote access utility and even perhaps collect data live yourself. So that's obviously not uniformly applicable, but in many cases that is available. So don't be discouraged that just because the travel and border restrictions might still be in place in some cases, you are prevented from using our infrastructure, because in most cases that's not the story. So we currently have about 64, I think there are probably a few more since this slide was made, of our Instruct services that are available for remote access. So it's worth exploring that. So I don't think any talk about structural biology in the last year or so could get away without mentioning the SARS-CoV-2 virus. And it's probably important and useful to note here that Instruct has made quite a significant response to the COVID-19 pandemic itself. So a lot of our services are very cutting edge services. They are in the moment of advanced technology delivery. And so we have been involved in a number of important advances that were relevant to the pandemic response. And I've just picked out a few of them here. The first on the left is our Instruct Centre in Belgium, in Netherlands, at Niesen, which is the electron microscopy centre. They were involved in a collaboration with Janssen Vaccines, and they helped to develop a stable form of the SARS-CoV-2 spike protein. And the stable closed conformation of the spike complex was shown to elicit more potent neutralizing antibodies, which of course was relevant to the vaccine design. And this work was done with Instruct support. So that was one example of work that had very important consequences for successful vaccine design. The second example in the middle there is work that was done in a number of Instruct centres actually, but I suppose mainly in the UK in this instance. This work that I've picked out came from the UK centre. And this was to map the antigenic anatomy of the spike protein against its receptor binding domain. And this mapping was done using a panel of 377 monoclonal antibodies to the spike protein. And these were found to bind most potently to the head and neck region, which is this region at the top of the spike protein around here. So that identified the major neutralizing antibody epitope regions on the spike protein, which again is important for vaccine development. And then as the pandemic got going and neutralizing antibodies started to be identified and collected from immune sera, then and the first of the variants appeared, the alpha variant appeared in the UK. And so the variant mutation of alpha was thought to make the neutralizing antibodies that had been elicited to the vaccine less effective. And this was structurally characterized, again, looking at the alpha variant structure of the spike protein and looking at the binding of neutralizing antibodies derived from convalescent serum in people that had had the wild type virus infection or had been vaccinated. And again, this has all been very important in understanding how the vaccine works and how to respond to new variants as they arise. And Instruct was integral to all of these studies. So as well as doing the scientific work, supporting the scientific work, we also were approached to help in developing a resource centre for SARS-CoV-2. And this included making available publications as they came out, some scientific breakthroughs as they came out, and they were very quickly published and made open access to everyone. But also it included building and making available a protein portal. And this allowed that information to be made available very quickly, and pointed people to secondary information accessibility as well. So our resource centre is still on the website. You can still see what's going on in there. There's work going up all the time. We haven't stopped our work on COVID-19 yet. We currently have around 18 major SARS-related publications that acknowledge Instruct, and more will be coming through. Some of these have been very high profile in Nature and Cell publications. So if you're a researcher though, and you're not working on SARS-CoV-2, one of the few who are not, what else can Instruct offer to you? And one of the things that we do is training. We have a very strong training program. In the last year, we've had a program that comprises of 7 training courses. There actually is another one in there, which I've highlighted there, coming up next week, which is the Best Practices in Cryo-EM, which is a virtual training course. Registration, I believe, is still open up until the 17th of October. Because it's a virtual course, you can continue to register. and participate in that. These have been very popular. This is an iteration of this workshop that has been oversubscribed before and is very popular. But we do also courses in all of the areas of technology that we offer. And we train people also with collaborative input from other research infrastructures or other facilities and interest groups or bodies themselves. For example, there's one there that involves FEBS as well. So training is one of our major events. And again, we try to concentrate on training in the latest technologies and their application to research. We also host a webinar series. So we have our Structure Meets Function webinar series, which is very popular. We have them monthly. And to date, we've had very good attendance. Almost 1,600 people have attended our webinars. From a very broad representation of countries, not just in Europe, but worldwide. So again, you can find all the details of the webinars coming up shortly. The last one last week was by Slovakia, and I think Lithuania will be on the next one to go. So I recommend you listen to these because it gives you an insight into the sort of science that is being supported in any of the Instruct centres and also the science that is ongoing in our members, and their scientific interests are very important to us as well. So apart from our own membership in Europe, as I said, we have very broad reach into lots of other areas, both other research infrastructures in different domains, but also we have participation and membership in a number of European groups and bodies. We are members of ERIC Forum. We until recently, chaired the Life Science Research Infrastructure Strategy Group, which is a European group of life science research infrastructures that consider issues like common policy areas, standards, and shared practices. We are integrally involved with ESFRI, which is the overarching policy group associated with developing the research infrastructure landscape across Europe. So we have lots of integration at different levels across Europe. And of course, this has meant that we have virtual meetings just about back to back every day. And we have struggled over the last 2 years to avoid virtual meeting fatigue. But it certainly has been very useful to be able to keep working and keep these communications open and the work going forward without being able to do these Zoom meetings, as tedious as they get to be sometimes. We wouldn't have been able to keep the momentum up for our programs in Instruct. So something that might be of interest to people is internships. And we have an internship program, which again is very popular. And internships provide funds for researchers, usually early researchers, at the beginning of their career path, to visit an Instruct centre for 3 to 6 months to do a research program or a research project associated with their own research interests. And internships are a very good idea for you to learn a new technique, to visit a different research environment. It broadens your horizons. It gets you to know a little bit more about how other people work, new approaches, new thinking. So it's very good for early stage career progression. And internships, as I said, are very popular. We have one call every year. And this year we have approved 5 internships. And that's about average for each year. We usually have between 4 and 6 internships approved each year. So these are nice things for young researchers to get involved in and experience a little bit of a different research environment, and in the process learn an awful lot. We also have a program, an award program, research and development award program, which supports, again, mostly early career research scientists, but is really not restricted to them. It's open to anybody who is at the early stages of developing a research project, and they need a little bit of introductory support just to get some preliminary data that might be required to go through a more conventional and expansive proposal for funding later on. So our research and development projects typically take about a year. They're done in your own home laboratory. You don't need to travel anywhere. But we support you to develop some preliminary data that will be very useful to attract further funding, and to do this using our infrastructure. So as part of the Research and Development Award, you get access to Instruct infrastructure for your experiments as well. And again, these are very popular. They're always heavily oversubscribed. We have an approval rate of between 10 and 15%. But again, they're very useful for those projects that are in their early stages, and they're sort of kickstart awards that could be very helpful. So that's sort of been a snapshot of all the things that we do within Instruct. But we also are involved in many European Union funded projects, and these cover a wide variety of different areas of structural biology input, but they extend way beyond structural biology and into some of the areas that are shown here. EOSC Life is looking at providing and managing and sharing data derived from life science research infrastructure outputs with the science community in general, using the European Open Science Cloud as the vehicle to do that. So I'm sure most of you will know that the European Open Science Cloud is in development right now, and EOSC Life was an EU-funded project to allow the life science research infrastructures to work out the processes and workflows to get life sciences data into the cloud, and to get those management processes sorted out before it becomes too difficult to do so. So Instruct is involved in EOSC Life. It's involved in another project related to EOSC called EOSC Future. It's an observer member of the EOSC Association, so it's integrally involved in developing the European Open Science Cloud that will be fit for purpose to manage life science data. And we're also involved in a number of other projects shown here. INEX Discovery, which is another project that deals with structural biology transnational access, but only for projects that have a translational application. ERIC Forum I mentioned, which is a group that brings together all the research infrastructures to consider common areas of practice and standards and policy. RIVIS is a project that Instruct leads that is increasing the visibility of research infrastructures so that people know what we can offer and the benefits of the infrastructure that we provide. And Transvac is a project that is there to develop procedures for vaccine development and translation into industrial application. So they're the ones that we have been operating in through the Horizon 2020 program, which is now at an end. And we're now into Horizon Europe, and the first round of calls for applications started this year. We went in for some of those. So we've already had 3 of the projects that we are participating in the first work program of Horizon Europe funded. So Isidore, BiCOVID, and EOS Future are all funded. Isidore is a COVID-related transnational access project, and BiCOVID is associated with it to manage the data arising from COVID-related research and manage that with links to EOSC. And then we still have another 4 applications through the Horizon Europe program in review at the moment. So those evaluations will be coming out later this year, and we hope if they're successful, they will come on stream next year. But you can see that Instruct is very well represented in European funded projects. And as a member of Instruct, then there's a very high likelihood that you would get some participation in some of these projects as well. So there is always the opportunity to get involved in some of these projects as a member of Instruct. And we also have international partnerships as well. So we have for some years had developed an international partnership with countries in Latin America. This was actually initiated many years ago in 2012 with a very small grant that we got from the British Society to develop interactions between the UK and Uruguay. And following from that, it then developed into something that was much more expansive and now includes Brazil, Argentina, Uruguay, Venezuela as well, and has attracted some co-funding from a number of projects, such as the Microbes project, which was based in Portugal, and the EU LAC Resinfra project, which is an EU-funded project to make bilateral collaborations between European countries and Latin American countries. So this now has developed in terms of Instruct's interest in Latin America to develop the structural biology communities in those countries in Latin America, and to be engaged with Instruct infrastructure in Europe as well. So this has been a really good bilateral partnership. Instruct has benefited a lot from understanding the science that is being done in Latin America, and we have been able to provide a pilot access program for the University of Sao Paulo, but also some of the scientists in Montevideo in Uruguay have also made use of Instruct services to understand the process, how research infrastructures work, and how to best interact in that way. So international partnerships are very useful for us, and a very good part of the work that we're interested to support. And we actually have, just go back one, we actually have, apart from connections in Latin America, we also have connections in South Africa. We held a structural biology workshop there last year, and we have good connections there, which are developing now. And we also had some very productive meetings in India, where there is a consortium of medical research institutes in India that are very interested in being an international partnership with Instruct as well. So I've talked a little bit about the infrastructure that we provide and how we support researchers and the science that they're interested in doing. But I thought you might be interested in knowing what sort of impact our services could have to some science outputs. So, you know, in most cases, the proposals that we receive are for quite small, contained, specific research projects that are, of course, very important for the researcher and their own particular interests. But sometimes opportunities come along to be involved in some very high impact science with quite far reaching importance and impact. And I just picked out a couple to talk about today. One is about virus assembly. And this was a publication in 2020, published in Nature Communications. And this is about looking at assembly of viruses in the reovirus class. And these are quite structurally defined class of viruses, but the actual replication process and assembly process wasn't clearly understood. And in looking at some of the structural components by cryo-electron tomography, unexpectedly, the images captured an intermediate stage in assembly of the virus that allowed the construction of an atomic model that provided insights into that assembly, and that led to understand the possibility of a better understanding of the whole virus replication cycle. So it was just fortuitous that this particular image was captured in an intermediary stage that provided these insights. But this has now been replicated in a number of similar viruses and seems to be a paradigm for assembly in these kinds of viruses, and probably will translate into other virus classes as well. So this work was done in the Instruct Centre UK, and as I said, these sorts of discoveries can actually have quite significant scientific impact, and it's really good to be involved in that kind of work, high impact work. And the other one I just wanted to point out was work that originated in Cambridge, but was done at with involvement of a number of Instruct centres, including the Belgium centre that provides nanobodies and megabodies. And these nanobodies or megabodies are small proteins. They're antibody-like single chain molecule proteins that can be used. They obviously have exquisite specificity, as do any antibodies, to particular ligand binding areas. And in this particular case, people were interested in looking at the GABA receptor, which, as you know, is GABA receptors are major inhibitors of neurotransmitter receptors, and they're responsible for fast inhibition of neural signals. So they are key targets for therapeutic intervention in neurological diseases. And people in Cambridge were very interested to understand the structure of GABA receptors so that they could target areas with chemical compounds that might be inhibitory or modify their activity. And it has been very hard to identify the structure of GABA receptors in any detail because of their size, the fact that they're membrane bound, all sorts of difficulties. But megabodies allowed them to attach the megabody to the GABA receptor and then do some cryo-EM imaging. And the megabody attachment allowed the images to be aligned so that very high resolution structures could be determined by cryo-EM. And so here you have some of the structures that were identified with the help of megabodies attached to the complexes. And at the same time as the Cambridge group were working, which is the top example there, there was another group also looking at GABA receptor signaling mechanisms, and they also used Megabodies to reveal a link between the structure and function that allowed some of the targets for chemical compounds to be identified as inhibitors of the GABA receptor function. So again, you know, this work couldn't have been done without Instruct Center Belgium providing the Megabodies, and it couldn't have been done without the cryo-EM facility in the UK helping with collecting these images and processing them. The image processing, I think, was done with the help of the Spain Instruct Center and the software there. So this is the sort of impact work that can be possible using Instruct infrastructure. So I think that's all I was going to say. I'm just looking here at the Instruct-ERIC website, which says PRG and not ORG. So our website is actually instruct-eric.org. And I encourage everybody to have a look at the website, have a trawl through. There's lots of interesting information on there. Of course, you can submit an application through the website as well. But it's probably worth just having a look around. If you have any questions, you can always contact any of us at any time for advice or help, or just what do I do now? But some of those questions maybe I can answer right now. So if you want to put your hand up or put some questions in the chat, then please do so. I'm open for questions. Okay.
Yeah, I have a question.
Mm-hmm.
So thank you for the presentation. Since the projects, you know, which are in this program involve many, probably also different countries, I was wondering how it solved actually the IP issue, you know, all this intellectual property, because I mean, some techniques which are not available, let's say in Lithuania, would be done in other countries. So how actually internally this all the things are processed and resolved in this situation?
So that's a good question. So most of our research proposals that we provide infrastructure for are proposals that are from academic scientists who agree to eventually release their data to open access and publish in peer-reviewed journals. And our principle is that in all cases, if you go to an Instruct Center and collect data, the data belongs to you. It doesn't belong to the center. So that data is yours. There's no shared IP. And we, Instruct, will agree to fund that work if you agree that the data that you collect will ultimately be published openly. So we do have the opportunity to do work with industries where there are IP issues, but in that case, Instruct does not fund the work through our own sources of funding. Anybody wanting to do protected IP research would need to use a pay-for-service fee. But for normal academic proposals, which can be published and we pay for, then you own the data, we don't.
Can you also elaborate a little bit on the total budget of this program, and what would be, you know, let's say the smallest project, you know, which would be funded, and the highest in terms of amount of money, you know, which would be allocated to let's say in these particular infrastructures? I understand also that particular techniques or technologies, they are very expensive in a way, you know, especially, you know, all this X-ray and so on, the structural things. But in general, just to have a feeling, you know, what we are talking about here. Maybe you could give an example or something like that.
Well, you're right. I mean, the cost of the various services that we offer vary widely. You know, some of the biophysical techniques are really not very costly at all, and they don't take very long to do. Whereas at the other end of the spectrum, if you want to use some of the high-end, high-field NMR machines, or you want to do cryo-electron tomography, for example, these can be very costly, and you know, typically a visit might take 2 to 3 days, or even perhaps a bit longer for NMR. So they vary, of course, in cost from a few €100 to several €1,000s. In Instruct, what we do is we have a capped amount of funds that we pledge for any one experimental visit. And that cap will cover if you need the most expensive of our infrastructures. And so that covers cryo-EM, for example, some of the X-ray crystallography beamlines, and also NMR. But even then, the funds that we pledge for to cover the proposal is only a contribution towards the real cost that is being borne by the Instruct Centre. So all of our Instruct centres provide an in-kind contribution in providing the infrastructure to make it available for access to Instruct proposals, and to support that access with trained staff that will provide expertise. So we don't directly pay for the staff to help with your experiment. We don't directly pay to purchase the equipment or to maintain it. That is at the cost of the Instruct Centre, and they provide a proportion of that as an in-kind contribution, so that it allows Instruct proposals and researchers to access it and do the work. So we top that up. We partly compensate the centres for their provision of that access. And we also provide a little bit of money to the user if you need to go to the research centre to do the work. Then we will provide the user with some travel and accommodation costs, so that you can go there, you can do your experiment, and you can come home. But we do have to cap our costs, because, as you say, we could very easily then commit an enormously huge budget to a single experiment, and that would not be fair for other users. So we do have a cap of €5,000 that we will max out on any single proposal.
Maybe the last question then. At the beginning, you mentioned that, I mean, there are teams, for example, which provide some expertise or things like preparing some proteins, for example, developing expression system and then express this protein in particular, let's say, E. coli or mammalian system. But these could be, you know, already achieved. And are there any databases, you know, where I can look and say, you already done that, maybe I can use your resources, let's say some clones or some produced protein which you might have it somewhere, you know, or is it something like that that would be available, you know, to see what was you know, collected? Because I know that some of the consortiums, you know, they collecting, let's say, particular protocols, and they putting online, they putting already some clones or something which would be available even to the broader community at some later stages, not to repeat the same what was done previously, you know, to save some resources, because it makes no sense to do expression of particular proteins if somebody did it already, you know. It's better, you know, to take it over, you know. So
So that I think that would be something that you would discuss with the centre staff themselves. They may well have knowledge of similar or the same proteins that have been expressed either in their own system or elsewhere. Quite often our proposals that request protein production have not expressed their own protein, and they don't know which system would be the best one to use for doing that, and so the centre will advise on the best one and provide the vector. So they would send the vector to you. You would do the cloning and maybe some preliminary expression steps, and then if that's successful, that would be taken on as a proper project and developed and scaled up so that you get good quality protein of the right amount for the next step in your experiments. But it would be, we don't have a database of clones containing various fragments of protein. But I think, you know, it's always worth asking people at the centre where you're interested in doing the work whether they know of anything that is relevant there. But certainly, you know, if you decide to go ahead with protein production and you have your genomic sequence or gene sequence that you want to express, then in most cases they will provide you with the vector appropriate for the expression system that is best.
As we are running out of time, I would like to thank Dr. Susan Denke, the coordinator of the Instruct-ERIC Hub in UK, for such an interesting and detailed presentation. And I can see there are some questions unanswered, but we will answer in written. We can answer in written. We'll get some connection with you again. And one thing I would like to announce that in 1 month, literally in 1 month, on November 16th, we will have our own Lithuanian webinar where 2 professors from Lithuania will give a speech. This will be Dr. Gintaras Valinčius and Dr. Virginijus Šikšnys. So we will see you everyone in 1 month. And thank you so much again for presentation and for organizing this webinar. And of course, we look forward to cooperate more and more intensively.
Fantastic. Thank you very much. And we look forward to your webinar next month.
Thank you so much. Goodbye.
Bye.